Hello students, in last video we have seen the introduction of chapter 3 human reproduction, okay, and today we are going to start with the topic male reproductive system, okay. We will begin with the male reproductive system, and in that we will talk about testis first, okay. We will talk about testis, then we will talk about accessory ducts of the male reproductive system, and then we will talk about accessory glands of the male reproductive system, and in last we will discuss about the external genitalia. So we will begin with the testes first, okay. So testes are present in one pair. They are present in one pair and they are primary sex organ also. Testis is a primary sex organ also, and it is known as an endocrine gland also, okay. So testis is a primary sex organ as well as it is an endocrine gland also, okay. So it has two functions to perform. It works as primary sex organ also and it works as an endocrine gland also. So when it works as primary sex organ, it produces sperm, okay. When the testis work as primary sex organ, it produces sperms, and when it works as endocrine gland, it produces male hormones like testosterone, okay. So testis has dual function to perform. It is a primary sex organ also and it is an endocrine gland also. When it works as a primary sex organ, it produces sperms, and when it works as an endocrine gland, it produces male hormones like testosterone, okay. And it is located, what is the location of testis? Testis is located in the scrotum. Scrotum is a sac or scrotal sac. It is present in the scrotal sac or scrotum, which is present outside of the abdominal cavity. So testes are located in the scrotal sac or in the scrotum, and it is present outside of the abdominal cavity. And testes are 4 to 5 centimeter long and they are 2 to 3 centimeter wide. So length of the testis is 4 to 5 centimeter and its widthness is 2 to 3 centimeter or 2 to 3 centimeter wide, okay. Then what is the envelope of testis? Testis is surrounded by an envelope which is made up of connective tissue. The envelope which surrounds the testis is made up of connective tissue, and this tissue is known as tunica albuginea, or that envelope itself is known as tunica albuginea, okay. So tunica albuginea is the covering or envelope of the testis which is made up of a connective tissue. So tunica albuginea is a connective tissue which makes the envelope of the testis. Then when we take the cross section of the testis, we find that it has 250 compartments. It has 250 compartments, and these compartments are known as testicular lobules, okay. Say for example, this is testis is there, okay, oval in shape, and this testis, when we take the cross section of this testis, we find that 1, 2, 3, 4, 5 up to 250 testicular lobules are there inside the testis, okay. So this is the one lobule, this is second lobule, this is third lobule, this is fourth lobule, fifth lobule. In this manner, total 250 compartments are there, and each compartment is known as testicular lobule. So when we take the section of the testis, we find that it has 250 compartments, and each compartment is known as testicular lobule. Now each compartment has a highly coiled tubule inside. Each compartment has highly coiled tubule inside, and the number of this tubule can be 2 to 3, okay. So 2 to 3 highly coiled tubules can be present in the testicular lobule. See, say for example, this is a tubule is there which is highly coiled in nature. So 1, 2, 3, 1, 2, 3, 3 in number. They are 3 in number, or there can be 2 in number also, okay. So in this manner, highly coiled tubules are present in each testicular lobule. Highly coiled tubules are present in each testicular lobule. So when we take section of the testis, we find that it has 250 compartments. Each compartment is known as testicular lobule, and each lobule has 2 to 3 highly coiled tubules. Each compartment is known as testicular lobule, and each lobule has 2 to 3 highly coiled seminiferous tubules. Okay, so this structure are known as seminiferous tubule. This structure is known as seminiferous tubule. It is highly coiled in nature. It is highly coiled in nature. And sperms are produced inside the lumen of this tubule. See, these blue colored tubules are there. Inside the lumen of that tubule, sperms are produced. So sperms are produced in the lumen of the seminiferous tubules. Sperms are produced in the lumen of the seminiferous tubules. So this is testis, and this testis has 250 compartments. Each compartment has 2 to 3 highly coiled tubule-like structure known as seminiferous tubule, and inside the lumen of that tubule, sperms are produced. So sperms are produced inside the lumen of the seminiferous tubules. Okay, so sperms are produced in the lumen of the seminiferous tubules. Okay, and outside the tubule, this white region which is present outside of the tubule, white colored region which is located outside of the tubule is known as interstitial tissue. Okay, white colored region located outside of the seminiferous tubule is known as interstitial tissue. And interstitial tissue has Leydig cell. Interstitial tissue has Leydig cell or interstitial cell, and these cells release hormone called testosterone. Okay, so outside of the tubule, the interstitial tissue is present, and in this tissue Leydig cells are there, and these cells are also known as interstitial cells, and these cells release male hormone called testosterone. So seminiferous tubules are present in the compartments. Seminiferous tubules are present in compartments known as testicular lobule. But outside of the seminiferous tubule, interstitial tissue is there, and that tissue is having interstitial cell or Leydig cell which will release testosterone hormone, and this testosterone hormone is essential for beginning the spermatogenesis or for continuation as well as beginning of the spermatogenesis. Okay, so spermatogenesis occurs under the action of the testosterone. Spermatogenesis occurs under the action of the testosterone which is released by the Leydig cell. Okay, now inside the tubule, another cell is also there which is known as Sertoli cell. Inside the tubule, another cell is also there which is known as Sertoli cell, and this cell nourishes the developing sperm. Okay, inside the tubule, Sertoli cells are present. These Sertoli cells will nourish the developing sperm. Okay, during the spermatogenesis, it gives nourishment to the developing sperms, and it gives nourishment to the developing male germinal epithelium. Okay, so Sertoli cells give nourishment to the male germinal epithelium as well as developing sperm, and they are located, these Sertoli cells are located inside the seminiferous tubules. So we have talked about two cells, one cell which is located outside of the seminiferous tubule and one cell which is located inside of the seminiferous tubule. The cell which is present outside of the tubule is known as Leydig cell, which releases testosterone, which starts spermatogenesis. Okay, so spermatogenesis begins under the action of the testosterone, but during the process of spermatogenesis, the sperms need nourishment. Developing sperm need nourishment, and this nourishment is provided by the Sertoli cell. So Sertoli cell is also essential for spermatogenesis. Sertoli cell is also essential for spermatogenesis. Okay, now say for example, in this seminiferous tubules, sperms have been produced after spermatogenesis. Inside this seminiferous tubule, sperm has been produced after spermatogenesis. Now these sperms will be released from these tubules. Sperm will get released from this tubule, and that process of release of the sperm from seminiferous tubule is known as spermiation. Say for example, the spermatogenesis has been completed inside the lumen of this tubule. Spermatogenesis is completed inside the lumen of this tubule. So then sperms will get released from the tubule and they will come out from the tubule. Okay, and that process is known as spermiation. So after formation, the sperms are released from the seminiferous tubules, and this process is known as spermiation. After their production, the sperm will be released from the seminiferous tubules, and this process is known as spermiation. So where will they go? Where will they go? The sperms will go into the accessory ducts. Okay, sperms will go into the accessory ducts of the male reproductive system. So now we will talk about the accessory ducts of the male reproductive system. We have talked everything regarding this testis. Okay, we have covered testis. So testis starts from here and it is completed over here. Okay, so testis point is covered, is finished over here. Okay, from this the accessory, from here the accessory duct, the topic of accessory duct will start. From here the topic of accessory duct will start. So let us revise the testis again. Testis is a paired gland, it is a primary sex organ also, and it is an endocrine gland also. When it works as primary sex organ, it produces sperm, and when it works as endocrine gland, it produces testosterone like hormone, and it is located in the scrotum, which is present outside of the abdominal cavity. And size of the testis is 4 to 5 centimeter in length and 2 to 3 centimeter wide. And the envelope of the testis is known as tunica albuginea, and it is made up of connective tissue. And if we take the section of the testis, then we find that it has 250 compartments, and each compartment is known as testicular lobule. So this is testicular lobule, and each lobule has 2 to 3 highly coiled seminiferous tubules. Each lobule has 2 to 3 highly coiled seminiferous tubules, and sperms are produced inside the lumen of the tubule. Sperms are produced inside the lumen of the tubule, and outside the tubule, the interstitial tissue is there, and in this interstitial tissue Leydig cells are present, and these cells will release testosterone. These cells will release testosterone, and testosterone initiates spermatogenesis inside the tubule. Okay, testosterone initiates spermatogenesis inside the tubule, and inside the tubule another cell is also there known as Sertoli cells, and these Sertoli cells give nourishment to the developing sperm during spermatogenesis. And when the sperms are produced inside the seminiferous tubules, they will get released from the tubules after their formation, and this process is known as spermiation. After their formation, the sperms get released from the seminiferous tubules. This process is known as spermiation, and they will come out in the accessory ducts. Okay, so when here the topic of the testis is over. One more point is left, that is the temperature of the scrotum. Temperature of the scrotum is generally 2 to 2.5 degrees Celsius less than the body temperature. Our average body temperature is 37 degrees Celsius, but the temperature of the scrotum is 2 to 2.5 degrees Celsius less than the body temperature. Why? Because this temperature is needed for the process of spermatogenesis. This temperature is needed for the process of spermatogenesis. So scrotal temperature is generally less than the body temperature. How much? 2 to 2.5 degrees Celsius less. Okay, and this temperature is essential for spermatogenesis. This temperature is essential for spermatogenesis. Now the sperms will get released from these tubules, and they will come out. They will come out in this network of the tubule, which is known as rat testis. After spermiation, the sperm will enter into a network of tubule which is known as rat testis. So here there is a network of the tubule is there which is known as rat testis. Okay, this red colored network is known as rat testis. So when the sperms are released from the seminiferous tubules, they will enter into the rat testis. They will enter into the rat testis. And what is rat testis? Rat testis is nothing but a network of the tubule. And from that rat testis, they will enter into a vasa efferentia. This duct is known as vasa efferentia. Vasa efferentia leaves the rat testis. Vasa efferentia leaves the rat testis. And vasa efferentia comes out from the pole of the testis. It will come out from the pole of the testis. Okay, so vasa efferentia will come out from the testis. Okay, so here seminiferous tubules are there where the sperms are produced. Then the sperms are released into the rat testis. From there they will go into the vasa efferentia, and vasa efferentia will come out, and then it will form epididymis. This duct is known as epididymis. Up to here, from here to here, it is known as epididymis. This duct is known as epididymis. and this epididymis covers the testis on the posterior surface. This epididymis covers the testis on the posterior surface. So this is rat testis. This is known as rat testis. This is rat testis. This is vasa effrontia. This is vasa effrontia, and then this is known as epididymis. Epididymis. Again, epididymis is also a highly coiled structure. Epididymis is also a highly coiled structure is there, and this epididymis covers the testis on the posterior surface. Epididymis covers the testis on the posterior surface, and then this epididymis gets converted into vas deferens. It gets converted into vas deferens. This duct is known as vas deferens. This is known as vas deferens. Okay. So after spermiation, sperms will go into the rat testis. Then they will enter into the vasa effrontia. Then they will enter into the epididymis. Then they will enter into the vas deferens, and then this vas deferens will travel through the inguinal canal which is present in the thigh. Then the vas deferens, sorry, this is vas deferens. This vas deferens will travel through the inguinal canal located in the thigh, and in that inguinal canal, vas deferens is known as inguinal duct. It is known as inguinal duct. So in the inguinal canal, vas deferens is known as inguinal duct, and then it enters into the abdominal cavity. When the vas deferens leaves the inguinal canal, when the vas deferens leaves the inguinal canal, it will enter into the abdominal cavity. So what is the pathway of the sperm? Sperms are produced in seminiferous tubule. Then they will enter into the rat testis. Then they will go into the vasa effrontia. Then they will go into the epididymis. Then they will go into the vas deferens. And that vas deferens also passes through the inguinal canal of the thigh. Vas deferens also passes through the inguinal canal of the thigh. And when it passes through the thigh, when it passes through the inguinal canal, it is known as inguinal duct. And then after being passed from the inguinal canal, it enters into the abdominal cavity. Or after leaving the inguinal canal, we can say, after leaving the inguinal canal, it enters into the abdominal cavity. So here it leaves the inguinal canal and then it enters into the abdominal cavity. And in abdominal cavity, it will loop around the bladder. It will loop around the bladder. It will take a turn like this, and then it will loop around the bladder. Okay, it will turn like this around the bladder, or it will loop around the bladder. So sperms enter into the rat testis, then they enter into the vasa effrontia, then they go into the epididymis, then they go into the vas deferens. Then this vas deferens passes from the inguinal canal in the thigh, and then the vas deferens enters the abdominal cavity, and then it loops around the urinary bladder. And here the vas deferens is joined by a duct from the seminal vesicle. See here bladder is there. Okay, here bladder is there, and on the posterior surface of the bladder, these two structures are present, and these structures are known as seminal vesicle. These are known as seminal vesicles. Okay, so a duct will come out from the seminal vesicle. A duct comes out from the seminal vesicle, and vas deferens will get joined with that duct. Vas deferens will get joined with that duct, and then they will form ejaculatory duct. So when the vas deferens loops around the seminal vesicle or bladder, when the vas deferens loops around the bladder, then a duct comes out from the seminal vesicle. And here both of them will get joined in form of ejaculatory duct. Okay, so it loops around the urinary bladder, and here it is joined by a duct from the seminal vesicle, and then they form ejaculatory duct. So here ejaculatory duct is formed, and this ejaculatory duct opens into the urethra, and urethra passes through the penis, and then it opens into the urethral meatus. Okay, so it loops around the urinary bladder, and here it is joined by a duct and form a seminal vesicle. Okay, it will get joined by a duct coming from the seminal vesicle. So here the duct that comes from the seminal vesicle will join the vas deferens. The duct that is coming from seminal vesicle joins the vas deferens, and then they will form ejaculatory duct. And then this ejaculatory duct will open into the urethra. This is urethra. Okay, so ejaculatory duct opens into the urethra, and urethra passes down through the penis and opens into the urethral meatus. So these are known as accessory ducts of the male reproductive system. These are known as accessory ducts of the male reproductive system. Okay. So what is the pathway of the sperm? Initially they are produced in the seminiferous tubule, then they travel through the red testis, then they enter into the vasa efferentia, then they come into the epididymis, then they go into the vas deferens, then this vas deferens passes through the inguinal canal and enters into the abdominal cavity, and in abdominal cavity it takes a turn around the urinary bladder, and here it is getting joined by another duct coming from the seminal vesicle, and both of them will join to form ejaculatory duct. Okay, so duct from seminal vesicle and vas deferens join to form ejaculatory duct, and this ejaculatory duct opens into the urethra, and then urethra passes down through the penis and it will open into the urethral meatus. Okay, so this is known as accessory. These are known as accessory ducts of the male reproductive system. So we have covered two topics in this video. We have talked about testes and we have talked about accessory ducts of the male reproductive system. We are remaining with the accessory glands of the male reproductive system and external genitalia, but we will cover them in the next video. Today I am keeping up to this only. We are still remaining with the two parts of the male reproductive system. They are accessory glands and external genitalia. That we will cover in the next video. Okay, till then, goodbye and good luck.